Okay guys, so today we're going to go over our stages of mitosis. Last time we went over interphase, so just to do a quick review from last time. When the cell is not dividing, when it's not making a copy of itself during mitosis, it's in interphase. And we had four stages of interphase. We had G1, S, and G2. And then we had this other phase called G0, where the cell completely leaves the cell cycle and it's no longer reproducing. But we're going to concentrate more on those three, G1, S, and G2. So what happened there was G1, the cell grew after it just was a newly formed cell after cytokinesis. In S phase, our DNA replicated, so we had a copy of our DNA. And then during G2, was where we had all of our checkpoints to make sure our DNA was replicated properly. After that, after everything is good, DNA is good, we are going to go into M phase, which is mitosis. So there's four stages. There's prophase, metaphase, anaphase, telophase. We're going to start off with prophase here. And I gave you some different pictures to show you what these stages are going to look like under a microscope, and then more of an animated, easier to see picture so you guys can actually see what's going on instead of us looking at the microscopic level. So during this, what's going to happen, it says our DNA shortens. So what this means is our DNA during prophase goes from chromatin to a chromosome. So it gets condensed. So now our DNA is chromosomes. All right. Also, during prophase, our nucleus and the nuclear membrane is going to pretty much dissolve. Now, it can reform later on. It just breaks apart into little tiny pieces, and it goes throughout the cell. But this is to make sure our DNA, our chromosomes, they can move everywhere. All right, so far we got two things that happen during prophase. Our first one here is our chromatin becomes chromosomes, so DNA condenses. And our second one, the nucleus breaks apart. And here's what everything looks like. Just kind of like an animated picture of it. You guys can see here the dotted line that represents the nucleus kind of dissolving. All right, so it's breaking apart. So anytime you see a dotted line, that's what they mean by that. And also the other thing I wanted to show you here is on the outside, right here. All right, these here, I'll only write it on one side. These are our centrioles. And if you remember, whenever we went over all the cell structures, we talked about centrioles, and we said they play a role in cell division, but that was pretty much all we said. This chapter, we're going to learn actually what the centrioles do. Okay, so big thing for prophase. Okay, we're going to go to kind of three things. First thing, our chromatin becomes chromosomes. All right, so that's DNA condensing. Second thing, our nuclear envelope breaks apart and it'll disappear. And then if you want to talk about a third thing, these centrioles, they're going to move to the opposite sides of the cell. Now, why they move to opposite sides is they are going to pull the DNA apart later on, and we'll talk about that later. But they're going to be, like we have North Pole, South Pole. They're kind of going to be, for our pictures, East and West poles. All right, so they're going to be on opposite sides of the cell. So this is everything we just went over with the centrioles, guys. They're also called centrosomes. All right, so same thing there. So plants do not contain centrioles, and this was the difference between animal cells and plant cells as we went over in the past. Our plant cells, they're going to use a different method to separate their DNA. But we're going to concentrate only on animal cells for this chapter. Okay, so we're going to be using the centrioles all the time. And then we already talked about them moving toward the opposite poles. So we're good there. All right. So what's going to happen here, guys, is our centrioles, and again, we went over some different cell structures a long time ago. We went over the cytoskeleton and we talked about microtubules. What's going to happen is these centrioles are going to, almost like Spider-Man, they're going to do like a web of microtubules, and those microtubules are going to attach to our DNA in the middle. Okay, so here is our second phase of mitosis. So prophase we're done with, all right? Pretty much all that happened there, our chromosomes condensed, our nucleus disappeared, our centrioles moved to the side. Now we're going to go to metaphase. And if you notice, our chromosomes here are lining up in the middle of our cell, okay? So the way I always think about this one, guys, is metaphase, the M, think of it as middle. Metaphase, middle. Our chromosomes line up in the middle of the cell, all right? Also, what's going on here is, if you look, here are our centrioles right here. Here's our chromosomes in the middle. All right. These are the microtubules I was talking about earlier. So they are going to go and they're going to connect to all of our chromosomes in the middle. Now, these ones over here are going to attach to these sister chromatids on this side. And if I do the other ones a different color, these ones on this side are going to attach to this sister chromatid on this side, okay? It doesn't matter which one they attach to, they'll just attach the one that's closer to them. Don't forget, guys, these sister chromatids are identical, so it doesn't matter which one goes to which side of the cell. We're gonna have the same DNA on each side, okay? And then the other thing with these microtubules I just wanna show you is, if you notice, we have some microtubules going to no chromosomes, okay? Here, what's going on here is they're anchoring themselves to the cell membrane, okay? So along with them connecting to the chromosomes, they're also anchoring themselves. And during anaphase, the next phase, I'll show you why they do that, okay? So metaphase, this is everything we went over. All of our karyotypes that you guys see with all of our chromosomes very, nicely depicted in them, all of those karyotypes, the chromosomes, the pictures for them are taken during metaphase. Why it's during metaphase is because our chromosomes are nice and condensed during that time, so they're very easy to visualize and see under a microscope. And here are some microscopic pictures of metaphase, okay? So you guys can see all the chromosomes are lined up right in the middle of the cell. It's pretty easy to depict there. And here is the non-microscopic picture. So you guys can see our centrioles and everything is going to those chromosomes in the middle. All right. I'm going to show you one thing with this picture, okay? Now, we're going to worry about this chromosome right here and this chromosome right here, okay? So we're going to worry about this one here, and I'm going to make the other one, we'll make it like a baby blue color, okay? We'll do this one here and this one here. All right, so just like get a mental image of what's going on here, just these two that I am pointing out to you. All right, right here. Okay, so I'm going to draw you guys a little picture here. So if you remember our karyotype, we had chromosome number 1. And we talked about the one from mom and the one from dad. Oops. And this is why I made those that certain color on the last slide. So now our DNA replicates. Okay, so now we have our 2 sister chromatids there from dad and our 2 sister chromatids there from mom. Okay, so what goes on, if I kind of try to redraw metaphase here real quickly, what goes on here is, and I'm not going to draw all of our chromosomes here, but I brought this one down and that one down. Okay, so let me draw my centrioles here on the side. I'm not going to do great pictures. And then I'll draw my microtubules going to the middle. Boom, boom. Boom, boom. Okay. So in reality, guys, because we have 2 X's for chromosome number 1 alone, and don't forget, it goes from 1 all the way to 23, 23 being our sex chromosomes. Okay. Right now, we copied all our DNA. Okay, so originally we have 46. So we had 46 altogether. After DNA replication, we actually have 92 chromosomes altogether. Okay, and each one of these is like 2 chromosomes right here, or we could say they're sister chromatids, same thing. Each one of these contains 2, so we would have 46 X's going straight down our middle line for metaphase. Okay, I just wanted to show you guys that kind of nice picture of it. All right. Okay, so the next one is going to be anaphase. Now, what's going to happen during anaphase is we are going to make sure that all of our microtubules are attached to all of our chromosomes in the middle. Okay, once we get the go ahead that, hey guys, we're all attached, we're ready to go, give me the green light. Okay, what's going to happen is those centrioles on the side, they are going to act like a fishing pole. So what the centrioles did from the side, they cast out their microtubules. They got their microtubules locked on to a chromosome. They made sure that the fishing pole, or the centriole on the other side, got its microtubule attached to that same chromosome on the other side, so that way we could pull that apart and the sister chromatids would separate. So now what we're going to do is, since we're connected with that fishing rod, with our centrioles, with our microtubules, we're going to reel those chromosomes in. And that's what happens during anaphase. Okay? So this is a microscopic picture of anaphase. Okay, now this one, I know we've been looking at it like, you know, this microscopic picture is shifted that way. So it's like north and south pole. That's where the centrioles are there. Here is an even better picture. So you guys remember that during metaphase, we had them all lined up in the middle here, real nice. Okay. What happened here was they separated. So we got half of our X coming to this side, half of our X coming to this side. And they're going to be reeled into the opposite poles of the cell. All right, so that is anaphase. So let's just do a quick review here, because we're a lot of information coming at you. We got prophase. So prophase, first step of mitosis. First step of, we also call it M phase. During prophase, 3 things. Our centrioles moved to the opposite poles, our nucleus disappeared, and our DNA condensed into chromosomes. All right. During metaphase, all of our microtubules attached to the chromosomes that were now lined up in the middle of the cell. Anaphase, we now took those chromosomes, or the sister chromatids, all right, and we separated them to opposite poles of the cell. So that's anaphase. And you guys can see here just slightly, right at the top and right at the bottom, we're starting to like pinch inward, okay? What this is doing is trying to start to create 2 cells. And then our last one is going to be telophase. So the chromosomes, they have finally reached opposite sides of the cell. They are at those opposite east and west, or north and south poles, depending on which way the cell is oriented. Okay. What's going to happen here is our chromosomes are once again going to uncondense. So they're going to go back to how they were during interphase and kind of at the beginning of prophase before they condensed. So they're going to once again turn into chromatin, and then the nucleus is going to reform. So telophase is pretty much the cell going back to how it was. And you guys can see here, kind of, I don't know, it looks like some kind of mutant character out of like a Marvel movie or something. I don't know. I just see like the bloodshot eyes and the little eyeballs in the middle. But anyway, you guys can see that there's like 2 cells here. Okay. They're not separated yet, but they're pretty close to it. So that's telophase. We get 2 cells, and you guys can see there's 2 nuclei in there as well. If we want to get with the animated picture of it, okay, and you guys can see that that cell is pinching inward a little bit more, okay, and once it's done pinching completely, we're going to have 2 new cells. These cells, guys, each have 46 chromosomes, and they are identical to one another. The DNA is 100% identical, all right? Following this, we're going to have cytokinesis. And remember, this is what we talked about before we went on into interphase, because the new cells were coming out of cytokinesis. All right. Now, for animal cells, best way I can describe how the cells separate in animal cells. Picture a balloon. Okay. I know this kind of looks like a peach, but this is actually a microscopic picture of it. What's going to happen is, picture a balloon, tie a string around the middle of the balloon, and start to pull that string tighter and tighter and tighter and tighter and tighter. So it's going like this, and it's slowly going in as we pull that string tighter. And then, now I know the balloon would pop, but if it wouldn't, okay, we could actually pull the string so tight where the 2 would separate. We would have 2 new cells. In animal cells, we call this a cleavage furrow. That is how it happens in animal cells. So it's, like a balloon with a string wrapped around it. That's only animal cells. For plant cells, it's a little bit different. We're making a new cell wall for plant cells. So what it is, picture like a piece of cheese or whatever. I don't know, like a brick of cheese, if you've ever seen a brick. Put it on a cutting board, take a butcher's knife, and just pow, straight down. All right. That is called the cell plate in animal cells, where we're actually separating those 2 cells just in one quick thing. So there's no like indent like the cleavage furrow in animal cells. For plants, it just boom, straight down the middle, and we get 2 new cell walls. It'll separate right down the middle, all right? And what's nice is it'll separate right where a new cell wall is forming, okay? And then overall, guys, once cytokinesis is done, okay, once cytokinesis is done, we are going to get 2 brand new cells, and we call them daughter cells. Both of them are daughter cells. So the original cell, the original DNA is still there, okay, but it's no longer like a parent cell. We call it them both daughter cells. So the original cell is still there, the DNA is still there, but we call them daughter cells at the end. All right. So we'll go through this just for a few minutes, guys. Just like if you're making cars at a factory, okay, sure, there's going to be a lot of machines that go into making the car. However, there's going to have to be someone at the end to make sure, okay, is this cold? Did the machine do this right? Like, someone's checking all the work the machines do. Obviously, the machines are going to work faster than humans, but the machines could still make mistakes from time to time. So we're going to have some checkpoints to make sure everything is good. So during G1, so interphase G1, the first checkpoint is during that part of interphase. And what we're going to do is toward the end of G1 is we're going to make sure that cell that just left cytokinesis, because they're small at the end of cytokinesis when they are 2 new daughter cells, we're going to make sure it's grown to where it needs to. All right. If it's not ready for that cell cycle, we're going to send it to G0. So that's our first checkpoint. Our second checkpoint is going to be during G2. So S phase, between G1 and G2, we are just going to replicate our DNA, and that is just, a feat in itself, so we are just going to worry about that. During G2, we're going to make sure all the DNA has been checked and replicated properly. And I'm not going to get too far into this because we got a whole chapter on this exact thing. Just this slide alone is its own chapter. So, yeah. And then if the cell, this is our last one, guys, so we have 3 of them. If the cell gets through G1 checkpoint, G2 checkpoint to make sure the DNA has been replicated properly, there is also a mitosis checkpoint. All right. During the mitosis checkpoint, guys, all we're doing is making sure all the DNA is lined up in the middle, and that DNA is connected to the microtubules. All right. Okay, guys, we'll stop here and we will start to go over cancer next time. So have a good rest of your day. I'll see you next time. Bye, guys.